The last part of the integumentary system to cover are the appendages of the skin. All of these appendages that we're going to talk about, your hair and the hair follicles that they're growing in, your sebaceous glands, the sweat glands, they're all considered derivatives of the epidermis. During embryonic development, what happens in the epidermis is some of the cells within that covering of the embryo begin to fold inward. They invaginate and they burrow down into the developing dermis at that time. Now, most of these structures do end up reaching the surface of the epidermis, either directly or indirectly, but they definitely all originated from the primitive epidermis that was once existed as an embryo, which is what makes them all epidermal appendages. As such, they are all going to be made of various epithelial tissues, just like the epidermis is an epithelial tissue. So let's start with the nail. That's what we're looking at here at the bottom of this picture. The nail contains, it's basically a modification of the stratum corneum located on the posterior distal surfaces of all of our digits for the fingers, and of course located on the dorsum surface of the foot at the distal edge of your toes there. So your nails are there to protect these potentially exposed appendages, right? The tips of your fingers and the tips of your toes. So this really strong stratum corneum is basically a bunch of dead keratinocytes. They make a slightly different version of keratin, the keratinocytes that become the nail. And so what happens if you take a look at the bed of the nail here, so the nail bed, or really the root of the nail, is going to be located at the proximal most part of the fingernail. There's a group of living cells here, basically the stratum basale cells that are specialized. And what makes them so special is that they are dividing like crazy. It's called the nail's matrix. These matrix cells in the nail are dividing. These are giving rise to all of the keratinocytes that end up dying and becoming these sacs of keratin in the actual nail itself. Of course, the keratin is slightly different than the keratin of your skin, and that's what results in the hardened surface of the nail, making it different than the stratum corneum of your skin. We have the root of the nail embedded just deep to the skin at the proximal edge. We have the body of the nail, that's the part of the nail that's growing out over the surface of your finger. And then we have the free edge of the nail that grows out beyond the surface of your fingertip. When you look at the body of the nail, you'll notice that it has kind of a pinkish appearance to it. And that's because the nail itself is this whitish, kind of translucent appearing structure. The pink coloring is coming from the dermis that you're seeing through the nail body. The dermis down below or deep to that nail body contains blood vessels. So the color that you see is due to blood flow through that dermis. This is why sometimes your fingernails can look kind of purplish if you're really cold, because remember, those dermal blood vessels will constrict, reducing blood flow through that area, showing a more or a less pink and certainly a more purple color in that fingertip there. So that's what you're seeing through the nail bed. It's not actually the true color of the nail. The nail itself is this whitish, somewhat translucent structure, coloring. That's the free edge part. The other part of the nail to point out is the eponychium. The eponychium is your nail's cuticle. This is a little bit of the neighboring epidermis that gets drawn out over the nail here, the surface of the body. So the eponychium is just a little bit of epidermis that gets drawn out. The lunula or lunule part of the fingernail you're seeing here, it has a little bit more of a whitened appearance to it, a little less pink here. That half-moon lunula shape that you're seeing at the proximal edge of the nail comes from the thickened stratum basale located deep in this area of the nail. So it is less translucent here. You're seeing fewer of the blood vessel colors from the dermis down below. As the stratum basale thins out, that's where you start to see through the nail's body, and you can see the color of the dermis down below. Okay, the next structure is our hair. The hair itself has a root, a shaft, and a bulb. So the shaft of the hair is the part of the hair that's exposed. You can see that superficial to the stratum corneum of your skin. The root of the hair is the part of the hair that's embedded in the skin. So the root is passing in through the epidermis, and it's also passing down here through the dermis of the skin. This is all the root of the hair. At the deepest part of the hair's root is the bulb. The bulb is the location where the hair is actively growing from. If you take a look under high power magnification under the microscope at a hair bulb, what you'll see is a little bit of dermis. So here the blue is staining collagen. You can see that the surrounding dermis, this is reticular dermis that we have down here, kind of bulges up like a little flame into the hair bulb. That little bit of dermis is known as the hair papilla, and it provides blood vessels to these hair matrix cells. The hair matrix cells are sitting right on top of that little hair papilla, and these are the cells that are responsible for dividing, and giving rise to all of these cells that eventually will die and become the actual hair itself. Hair is also a whole bunch of dead cells that are tightly packed together as protein sacs, just like the stratum corneum of the skin, just like the fingernail. So we've got all of these dead cells that end up packing tightly together as our hair. Whole bunch of protein, really, at the end of the day. But they were once living cells. If you look all the way down at the bulb, you do have many living cells down here that are dividing. Mitosis, tons of mitosis happening down in the bulb. One of the things to note is that the hair is actually growing through a tube called a follicle. The hair and the hair follicle are two separate structures. To make it clear, if I were to pluck a hair and I plucked it out and I got the root, and let's say I got all the way down here, I even got the hair's bulb, I would not remove the hair follicle. So you can pluck a hair, but you're still going to have the tube through which that hair used to grow. That will still remain right in place there. Hair follicles are just tubes through the skin. That tube contains an epithelial root sheath. So the hair itself is this pink area here. The epithelial root sheath is on the surface, right? This kind of inner surface of the hair's follicle. And then the connective tissue root sheath is the outer surface of the hair follicle. So this would be the hair follicle over here on this side, and the hair follicle over here on this side, an epithelial root sheath and a connective tissue root sheath. If you've ever plucked a hair and you get that white stuff, right, that you get all the way down at the base of the root and the bulb, that white stuff is actually the epithelial root sheath getting ripped out along with the hair. There's living cells in that epithelial root sheath, and that's why we can get DNA from a hair that contains living cells from this kind of a pluck. If you, on the other hand, just go for a haircut, maybe you shave the hair, right, buzz it off, there are no living cells on the shaft of the hair, so you wouldn't find any effective DNA. No nuclei in those dead cells are ever going to exist. So DNA from hair requires living cells. You can get that from either the hair's bulb, or you can get that from the epithelial root sheath that would be coming out with that hair. The last thing to mention are the erector pili muscles. Erector pili muscles are little strips of smooth muscle that anchor the hair follicle, not the hair itself. They're anchoring the hair follicle to the neighboring area of reticular dermis. These erector pili muscles will contract, and when they do, it causes the entire follicle to stand upright. Well, when the follicle straightens up, so does the hair within it, and that gives us the appearance of our hairs standing up on edge. So if you've ever experienced goosebumps or goose pimples, that's because of your erector pili muscles. Every single hair follicle has one, all contracting at the same time, all those hairs standing up on edge. This is one of those responses that we don't have conscious control over. It's something that our autonomic nervous system controls. So your hairs might stand up on edge when you're excited. They might stand up on edge when you're nervous. That is your erector pili muscle contracting. A little bit of smooth muscle there in the reticular layer of the skin. Next up are the glands. So these glands are also epidermal appendages. If you look at any gland, you'll typically find a coiled set of tubes anchored down deep low in the reticular dermis, with a long duct reaching either all the way up to the surface of the epidermis or potentially reaching to the surface of the hair follicle itself to secrete onto the surface of the epithelial root sheath within the hair follicle. These secreting glands come in different types. So we'll start with the sweat glands. Sweat glands are secreting mostly watery sweat, watery material. There's two different types of sweat glands that we find throughout our bodies. Your merocrine sweat glands, these are your true sweat glands. They're also sometimes called eccrine sweat glands. Eccrine is spelled with an E. These merocrine or eccrine sweat glands are typically going to be lined by simple cuboidal epithelial tissue. Occasionally we find a larger duct that's lined by stratified cuboidal, but remember that we'll never look at those stratified cuboidal tissues in this class. These merocrine sweat glands secrete a watery serous secretion. It's got lots of ions, sodium ions and chloride ions, but mostly water. That's our true sweat. The merocrine sweat glands are known for secreting directly onto the surface of the epidermis, and they are located everywhere. They're located in thick skin, they're located in thin skin, they are abundant throughout our bodies. And the main function of this merocrine sweat is to help cool the body. So this evaporation of this sweat helps to facilitate keeping your body cool. The apocrine sweat glands are a little bit different. You can see that the cells look a little bit different. They're also still simple cuboidal epithelial cells in your apocrine sweat glands, but they secrete a really lipid-rich and protein-rich secretion. So it's still water-based, but the secretion from apocrine sweat contains more proteins and more lipids. The apocrine secretions are often going to secrete into a hair follicle, and those apocrine secretions are going to, from there, percolate up to the surface of the epidermis. Well, up here on the surface of the epidermis, we do have a normal flora of bacteria, these kind of healthy, normal bacteria that live on the surface of our skin. Normally, their numbers are kept in check because they don't have an adequate food supply to really divide. However, when apocrine sweat arrives, it's got all these proteins and lipids, the bacteria basically have a buffet, and they start to really eat and use all those nutrient molecules to metabolize. When the bacteria metabolize the proteins and the lipids coming from this apocrine sweat, they release gases, and it's those gases that are responsible primarily for body odor that's associated with apocrine sweat. If you look at where we find the apocrine sweat glands, it's generally in what you would consider your smelly parts of your body's skin. The axillary region, the anal region, the genital region, the inguinal region, the areola, that's all going to contain these apocrine sweat glands, releasing again this nutrient-rich sweat for the bacteria to be able to feast upon. So apocrine sweat itself generally doesn't have too much of an odor. If you were to, go work out in the morning right away, you probably aren't going to, notice any sort of body odor. But after a little while, those bacteria have been feasting. That's when you start to really notice that body odor. Toward the end of the day, for example, or a few hours after a workout session. That's when the apocrine sweat glands, the sweat has been actually used by those bacteria, and you start to get the production of those gases. So those are your sweat glands. Again, merocrine and apocrine sweat. Merocrine is everywhere. Apocrine is just in a few select locations of the body. Next up are the sebaceous glands. Sebaceous glands look completely different. They are these sacs of, I always call them fluffy cells, but these are really just sacs of cuboidal epithelial cells, and they're located always hanging off of a hair follicle. always. These sebaceous glands secrete their substance into the inside of the hair follicle, and it percolates up to the surface of the epidermis from there. Sebaceous gland secretion is sebum, and sebum is an oil secretion. So this oily lipid secretion is associated with hairs because the sebaceous glands are associated with hair follicles. The sebaceous glands are going to secrete that oily secretion onto the surface of the epidermis in thin skin only. It turns out thin skin is the only skin of your body that contains hairs, and all thin skin contains hairs. It's more dense in areas like on your cranium, on the surface of your head, for example, and less dense maybe in other areas, maybe like the antecubital region on the anterior side of your elbow there. It doesn't have very many hairs, but they are there. It exists in all thin skin throughout your body. And so sebaceous glands are going to be associated with all of your hair follicles located in thin skin. Ceruminous glands, these are a specialized type of apocrine gland, actually. It secretes a really lipid-rich secretion known as earwax, and that's going to secrete into the auditory canal. That's your ear canal, the external ear canal. Helps to prevent infections. It helps to actually reduce bugs from flying into your ears. They don't like the ceruminous secretions. And then mammary glands, these are modified apocrine glands that develop during pregnancy. So men and women all contain mammary glands, but a combination of hormones that are secreted during a pregnancy stimulate mammary glands to begin to produce milk. And so when that mammary gland produces milk, that secretion can be released to nourish a newborn baby. This modified apocrine gland also produces a really lipid and protein-rich, kind of nutrient-dense milk to nourish that newborn baby. Okay, just a quick note on sebaceous glands. Sebaceous glands do secrete sebum or oily substance, and they do so by the entire cell popping. This is called holocrine secretion. The whole cell pops, and the entire secretion, including all of the phospholipid bilayers and organelles and nucleus, all make their way to the inside of a hair follicle. From the hair follicle, that sebum can bubble up all the way up to the surface of the epidermis of the skin. Sebaceous glands are responsible for acne. It turns out that sebaceous gland secretions are influenced also by hormones. And the surge of sex hormones that occurs at puberty really tends to stimulate these sebaceous glands to begin to really secrete an excessive amount of sebum. And that can cause clogs or blocked sebaceous glands and even blocked hair follicles. When that happens, we call this acne. There's a lot of different types of acne, but just to highlight a few of the more commons, all acne is a clogged sebaceous gland. If that sebaceous gland is completely clogged and covered, we call that a whitehead. If the sebaceous gland is clogged and covered, but there's bacteria that got trapped in there, you'll start to see a little reddened bump, and that's known as a pimple. So pimple is more of like an infected whitehead versus a whitehead is just a clogged sebaceous gland. And then a blackhead is a clogged sebaceous gland that is not covered, and so the sebum that gets blocked up in there oxidizes, giving a darkened appearance to the surface. So those are, you can think your sebaceous glands for the acne that results. And I know it says acne occurs in teens. It's a common time in our lives, but really any time where there are changes in hormone secretions, particularly sex hormones, can be particularly vulnerable times for acne development in an individual. In women, this occurs during pregnancy. Before, during, and after pregnancy, those hormone levels are fluctuating, and that can result in either excessive sebaceous gland secretion or super dry skin with reduced sebaceous gland secretion at any of those times. Women also experience menopause. The rapid reduction in hormone secretion at that time can lead to changes in sebaceous gland secretion. Men are not off the hook. Men experience changes as well. Again, at puberty, this surges of hormone secretion, but also at other times in their lives, men can experience hormone secretion or hormone changes that result in sebaceous gland changes. And late in life, oftentimes male sex hormones reduce late in life as well, resulting in changes in sebaceous gland secretions. The last two things to talk about in this lecture are problems with the skin. So in every organ system that we cover, I do like to highlight some of the more common problems that you might come across in your future medical professions. And for the skin, we're going to talk about burns and cancer. So burns are a leading cause of accidental death, and that's mostly due to improper burn treatment, leading to either rapid fluid loss or infection. Burns come in three types. So you can see in the picture on the right, the first degree, second degree, and third degree burns. First degree burns damage the epidermis, resulting in inflammation in the dermis. That inflammation results in the reddened appearance because of the dilated blood vessels that we're able to see through the surface of the epidermis here. When that's occurring, the ability to vasodilate and vasoconstrict is reduced. And so patients with a first degree burn are typically not at too big of a risk of fluid loss or infection. However, that first degree burn patient can be at risk of hypothermia. Think about, spending a day at the beach or a day at the park and you get a sunburn and you stay in the evening time. If you don't have a sweatshirt or something to keep warm, these blood vessels are still going to be dilated even though it's getting colder and colder outside. And as body temperature drops, these blood vessels remain dilated, resulting in more heat loss to the cooler environment around you. So bundle up, keep your patients warm, especially if they've got any type of burn and even a first degree burn. Second degree burns do damage the underlying dermis, resulting in blisters. So that's what characterizes a second degree burn. You do not want to pop a blister because that breaks the epidermis. And remember that the epidermis is a barrier to entry for microorganisms. So once you've broken that epidermis, a microorganism can potentially get into the body through that break in the epidermal layer there. So infection becomes a risk with second degree burn victims, and also fluid loss. The ground substance that's located in the dermal layers, the papillary dermis and the reticular dermis, could also ooze out through any sort of breaks in the surface of the skin. That fluid loss can ultimately result in lowered blood pressure, leading to systemic shock. And then, of course, we still have the hypothermia issue here with a second degree patient. So keep the patients warm, keep them covered up, and prevent that fluid loss and infection by keeping the epidermis as intact as possible. A third degree burn victim, that is not possible because a third degree burn victim has lost their epidermis. The burn has completely removed the epidermis and has damaged the dermis and often the underlying hypodermis as well. Without an epidermis at all, the ground substance in the dermis, papillary layer, reticular layer, and in the hypodermis can easily evaporate out to the environment. There is no more waterproof barrier here. So fluid loss is a major concern, an immediate concern for a third degree burn victim, and that's mostly why we end up doing skin grafts. A skin graft takes the epidermis from another region of the body and covers the open wound, essentially here, the open exposed dermis. It also quickens the healing time and minimizes scarring that could occur if the scar tissue would form to rapidly cover the broken skin here. Of course, we still have infection as a major risk with all of this exposed deeper tissue. All kinds of microorganisms could potentially get into the body's circulatory system directly through the exposed dermis and hypodermis layers here. Those are your burns. The last is cancer. Carcinomas are a very common form of skin cancer. These are skin cancers involving the keratinocytes of the epidermis. Basal cell carcinoma and squamous cell carcinomas arise from mutations in keratinocytes in two different regions of the epidermis. Basal cell carcinomas occur when keratinocytes in the stratum basale acquire so many mutations that they start to divide out of control and they start to grow into the surrounding dermis as well. When you have a basal cell carcinoma, it tends to have kind of a shiny appearance. Oftentimes it gets a reddened or a purple appearance, and it often looks raised. So that's your basal cell carcinoma. A basal cell carcinoma of the stratum basale cells does have a very high cure rate. Surgical removal of the entire tumor, if that's possible, if it's able to be gotten early enough, can completely cure the patient. That basal cell carcinoma has a very low likelihood of metastasizing, of spreading to other organs in the body. Not that it's impossible, but it certainly has a low malignancy rate, a low metastasis rate. Squamous cell carcinoma looks different. So down here you can see it has more of a reddened, scaly appearance. It originates from mutations in the stratum spinosum of the epidermis. And these cells, when they divide rapidly and out of control, they form another bulging papilla. This big kind of bulge starts to invade some of the dermis down below as well, but it has a slightly different appearance. Another tumor that has a very good surgical cure rate. The surgical removal of the entire tumor generally cures the patients from their squamous cell carcinoma. It also has a very low rate of metastasizing, very similar to the basal cell carcinomas. The last skin cancer is a melanoma. You can see this guy looks very different here. Melanomas are skin cancers of melanocytes. Now, when melanocytes begin to divide out of control, they release a lot of extra pigment, a lot of extra melanin. And so this tumor tends to look very, very dark, kind of a blackish, purplish tumor, looks raised and very irregular in appearance. Notice how the cells look kind of big and bulgy over here. There's this extra little bulge. The cells over here might be dividing more rapidly than the cells right here, for example. So we get this very irregular shaped tumor. That's a pretty typical melanoma. Melanomas are particularly dangerous and deadly because of their ability to metastasize so early on in tumor formation. When a melanoma is beginning to grow, when melanocytes are beginning to divide out of control, they are very, very quick to lose attachments in the epidermis, and they are very quick to begin to move down and invade the underlying dermis. Not as a mass, as a tumor that's stuck together, but they invade the underlying dermis as individual cells. And they actually begin to migrate through the dermis, and once they start migrating like that, they can jump into the bloodstream. When they jump into the bloodstream, that basically gives these cells a highway to every other organ in the body. So what ends up killing the patient is not the original melanoma in the skin, it's the new melanomas that begin to pop up from these cells that are traveling through the circulatory system. Some of the cells might be able to jump out in the liver and start to grow melanomas in the liver. Some cells will jump out in and around the brain, and we start to get melanomas growing in the brain. Some of the cells might jump out in the lungs and in other organs. And ultimately, when melanomas begin to grow in all of these other organs, it begins to impair the normal functioning of these other organs, and the patient ends up dying due to the melanomas that are growing, the organs not being able to do their normal physiological functions for the patient. Another dangerous part about melanomas is that not only are these metastasizing tumors difficult to detect, but they're also difficult to destroy. The melanomas are very resistant to our current chemotherapies and immunotherapy techniques. And so many melanomas that are metastasized are going to end up being lethal for those patients. I think there's something called a 50% survivorship rate for diseases. The 50% survivorship rate is a measure of how many patients, how long after original diagnosis do 50% of the patients still survive. And so for melanomas, that number hovers, it's a little bit different, but it hovers around 12 months. So that's a pretty rapid rate of decline. That means 50% of patients are not alive 12 months later. 50% of patients are. So one of the things to keep in mind is size in this one. A 6 millimeter diameter melanoma has most likely already metastasized. 6 millimeters in diameter is about the width, about the size of the eraser at the tip of a pencil. So kind of keep that visual maybe in mind when you're looking at your body checks. So definitely do some skin checks, check your skin, get somebody to check your back for you. A lot of melanomas are missed because we don't check our backs too often. And you're going to be looking for the ABCDE rule of thumb here. So asymmetry tells us that if something looks different on one side than it does on the other, it's asymmetrical. Cells are behaving differently in those two different locations, so we want to get that checked out. Any sort of border irregularity, any sort of waviness, right? That's definitely something to get checked out. It tells us again that some cells are dividing more rapidly than others. Certainly color, a darkened pigment like this purplish blackish color that we're seeing here, or even a reddened color, right? These are all signals that we've got something abnormal happening, and we want to get those things checked out by a physician as well. Diameter, 6 millimeter cutoff, right, is our cutoff for melanomas, but anything that has a large width is something to get checked out. And then even if you've had something checked in the past, the last letter here, E, for evolving. If you notice that you've got something on your skin like a mole and it changes, it looks different than it used to, it's grown a little bit, or it starts to look a little more irregular, or it feels different, it's a little bit more sensitive, a little bit more itchy, those would be times to get those things checked out, because moles and other types of normal body lesions on the surface of the skin could potentially change over time and evolve into a type of tumor. So anything that's changing is worth getting checked out again. And just because you notice these things doesn't necessarily mean that you've got a skin tumor or skin cancer, but it does mean that you want to get it checked out to make sure that everything is falling within the normal